So we are going to roll into our first session on red clover and legume research. And our moderator for this session is Damon Mee from New Hampshire NRCS. So I'll turn it over to Damon.
Good morning, everyone. Thank you, Jen. Yeah, I think we're going to kick it off today with this great research on red clover and legumes. And so starting us off is Brittany Davis. And I encourage you throughout the day, any of our speakers, if you go on Whova, their bios are there and available to read through and at your leisure. And so I'm really excited to hear what Brittany has to share.
Thank you for having me here today. I'm always excited for an opportunity to talk a little bit about the research that we do and share it with people that might find some use out of it or might have some ideas or things that they'd like to share with me about how that might apply in their systems or in their agricultural practices. And so my name is Brittany Davis. And today I'm going to share with you guys a little bit about our research looking at isoflavones and legumes as functional forages and feeds in sheep and cattle grazing systems. In these presentations, especially those that aren't in Kentucky, I like to spend a moment just to tell you a little bit about where I'm coming from. And so I work for the USDA-ARS Forage Animal Production Research Unit. We're located in Lexington, Kentucky. Our mission is really to look at strategies to improve the productivity, profitability, competitiveness, and sustainability of forage animal enterprises within the transition zone. We have 5 scientists and 5 technicians. Like most other USDA-ARS units, we have a variety of expertise present in our unit, and our job is to study that mission. And so my role in that team is as an animal scientist, predominantly working in the field. We're a pretty small unit, but we're co-located at University of Kentucky, so we do a lot of research with faculty there as well as extension, and also our Kentucky Beef Cattlemen's Association, which I'll talk a little bit about today. By training, I'm a ruminant microbiologist, and so I start every talk talking about why are ruminants so exciting and so wonderful at converting grass into meat. And so ruminants are special because when they consume grass out in the pasture, the first site of digestion is fermentation. And so there's this big, diverse microbial community in the gut that is the first place at which all of these nutrients are digested. And that's different than us or different than horses or pigs, where we see enzymatic digestion first. Notably, they have 2 sites of fermentation. So they have this rumen, this huge fermentative organ, and then they have some enzymatic digestion that occurs, just like what happens in our bodies. And then in the hindgut, we see another site of fermentation. And so when an animal consumes pasture, it's consuming a variety of nutrients, things like protein and fiber, which then go to the rumen microbial community, and they're digested by all these rumen microorganisms to produce different acids that the animal can absorb to meet their energy requirements. And so in grazing ruminants, as much as 70 to 80% of their blood glucose comes just from this fermentation. And so it's really important to optimize, and that's part of the story of what I'm going to be talking about today. So another concept I want to introduce right up front is the concept of functional foods. And everyone's probably heard of functional foods in human nutrition. Typically, they confer health benefits beyond basic nutrition because they contain bioactive feed components or compounds that can provide an added benefit. And so one example of this is something like fatty fish. We can eat a few servings of fatty fish a week, and because it contains special fatty acids, we can see a reduction in things like heart disease. So a very small amount in the diet to confer this enormous health benefit. Sometimes these functional foods are referred to as nutraceuticals or superfoods or food as medicine. So how does that translate into animal agriculture or functional feeds? So there's a lot of feeds that have been studied that contain bioactive compounds and meet this definition of functional foods, and that they confer an animal production or health benefit when they're consumed regularly as part of the ration and in a relatively small proportion. And so that varies depending on the functional feed, but it can be as much as 5% of the diet to as small as a handful a day of a compound. So there's lots of research that's been done on functional feeds and their application in different types of livestock production systems. And this is just an example of some of those functional feeds that are out there and are in marketed products like capsaicin in peppers. There's some mineral supplements with that. Hops contains beta acids. It makes beer, but also contains compounds that can confer some benefits to livestock. And things like garlic oil are already present. However, as people who are most interested in grazing, what about functional forages? How can we capture this functional nature in our pastures? As we know, ruminants evolved to graze diverse native pastures. And so these pastures allow the ruminant the opportunity to both select forages for their nutrient composition. They're trying to get crude protein and fiber and what they need to energize their body, but also based on this phytochemical landscapes. Plants produce phytochemicals, as we all know, these secondary metabolites, not so they can grow or they can reproduce, but to add benefits, so other added benefits like insect herbivory deterrence or protection from UV light. But when animals consume these compounds, sometimes they can have these enormous physiological benefits for the animal, whether that be with growth or reproduction or even health. So this diverse phytochemical landscape that's available in these native pastures was coined the chemoscape. So basically the buffet of phytochemicals that the animal can consume. And so by modifying this buffet of chemicals, we can modify the performance and health of our animals. So today's grazing systems don't look nearly as diverse typically, at least not here in central Kentucky in this picture. So we mostly have things like different cool season grasses and some legumes thrown in there. But again, opportunity to think about adding to this phytochemical landscape to benefit both the pasture and the animal. Again, I want to nod my head to lots of research that's been done looking at phytochemicals that can be utilized in these grazing systems. So obviously we have things like Serratia lespedeza or forage chicory, or today we're going to do a lot of talking about clover. All of these different landscapes have phytochemicals in them and have been studied. Why do we use clovers in pasture systems? Principally to lower nitrogen inputs in our pastures. We also know that adding clover adds some quality to our pastures. So it's high in protein, highly digestible. It adds forage diversity. And if you take all of this together, there's tons of research that has shown an improvement in animal performance when you add some legumes into your pasture. But what about a different role of these clovers in pastures as a functional feed or a forage? So legumes, most notably red clover, contain bioactive isoflavones. So these compounds are part of the flavonoid family. They're antioxidants and also phytoestrogens, and they confer several benefits to the animal when they're consumed. And so when they enter into the gut, they can act to change the ruminal environment and lead to growth promotion, which is what we're really going to be talking about today. And then when they're absorbed across the gut into the bloodstream, they can act on the blood flow of the animal, which has some added benefit. The concept of altering the rumen microbial community to improve performance has long been done. So we use a lot of antimicrobial growth promoters, or did use a lot of antimicrobial growth promoters in livestock production, things like Bovatec or Rumensin and cattle diets. So what is an antimicrobial growth promoter? It's an antibiotic or other antibacterial agent that selectively will kill different bacteria in the rumen, altering how those nutrients are utilized, and then allow the animal to gain more weight and improve their feed efficiency. So if you think about that in terms of something that we're more maybe akin to, the pasture and how we manage pastures, we have forage species within that pasture, grasses and legumes that we want to keep around because they're important for our animals' performance. And then we have weeds that we spray out with herbicides that we're trying to get rid of because they're decreasing the productivity of our pasture, and we really don't need them around to improve the forage species. In the rumen, there's the same concept. We have fiber-type forage species. In this case, we have bacteria that are adhered to a forage particle. These bacteria are particularly important in the rumen. We want to keep those around and support their growth. And then we have wasteful species like these methanogens attached to this protozoa here. Everybody knows methane emissions, right? We want to kill those archaea in the rumen. When we look at antimicrobial growth promoters conventionally and how they typically work, it's to change protein utilization in the rumen. And so this is a schematic that kind of illustrates how that works. So if an animal is not consuming any antimicrobial growth promoter, but is consuming dietary protein, that protein enters the rumen where it's fermented by this big bad group of bacteria called hyperammonia producing bacteria. And so these bacteria will take all of that protein, digest it really quickly, and produce an enormous amount of ammonia. And so that ammonia can be utilized for different functions with bacteria, but typically most of that ammonia is just excreted in the urine. So a good chunk of the nitrogen that's coming into the diet is just being wasted and put back out on our pastures, or at least not available for the animal. However, when you add in a ruminal antimicrobial growth promoter, which is that it selectively kills those bacteria that are wasting that ammonia, and therefore that protein that's coming into the diet will pass through the rumen and make it to the rest of the digestive tract where it can be utilized and absorbed by the animal to be used for growth and meat production. And by doing this small change in the rumen microbial community, we can really influence that animal's performance. Another reason why we are thinking about alternatives and phytochemicals for use in livestock production is obviously the veterinary feed directive. So we're trying to reduce the use of these conventional antimicrobial growth promoters and find some natural alternatives that can be utilized. So another important group of microbes in the rumen are those that use structural carbohydrates. And I referred to these earlier as our beneficial species or forage species. And so the enzymes that break down fiber are strictly microbial. The animal cannot break down fiber in its diet on its own if it didn't have these bacteria and microorganisms present to do so. And so supporting that community is very important if we want to think about animals that are grazing high fiber diets like those when they're grazing forage out on the pasture. However, some of these antimicrobial growth promoters that have been used historically actually kill these bacteria. And so there's been a lot of research evaluating some of these conventional antimicrobial growth promoters. And we often see, it's a busy table, but just looking at the up and down arrows, you can see there's a variety of results that can occur when you add in these ionophores into grazing systems. And so sometimes you see some benefit, sometimes you see no effect, and sometimes you see even detriment to the performance of animals grazing. And that may be linked to their effect on fiber utilization in the rumen. And so alternatives that don't impact fiber utilization are of interest as well. Spinning all the way back to our isoflavones, specifically biocanin A, our research group has spent about 10 years evaluating this isoflavone as a selective inhibitor in the rumen. And what biocanin A does is that it inhibits protein fermentation. It kills out those hyperammonia producing bacteria, the bad guys that I was talking about, but it promotes fiber utilization. So it doesn't kill those fiber utilizers. It acts by a different mechanism, and so they're not susceptible. This has been shown to result in increases in those acids that the animal can use for energy. And then we've also had some really promising benefits of applying these compounds in grazing ruminants. And so today I'm going to spend some time talking first about some of our beef cattle research on the subject, and then move into some of our small ruminant sheep research, more recent research on the subject, to tell you a little bit about how we can apply these in grazing systems. So again, what about pasture? So the first question we wanted to ask ourselves was, does it work? And so we set up this really simple grazing study where we had 3 different treatments. We had steers that were assigned to a pasture-only treatment, so just consuming cool season grasses out in the pasture. We had a dried distillers grain supplemented treatment where steers were consuming just dried distillers grains as a protein supplement. And so why we use dried distillers grains is it has no isoflavones in it. It doesn't have any special phytochemicals. So it's a good protein control. And then we had the same amount of dried distillers grains, and we just sprinkled some biocanin A on top, a very low level of biocanin A into those diets. And so when we look at average daily gain in this figure here, you can see that adding biocanin A on top of the dried distillers grains increases performance dramatically. And so we see a 29% improvement in performance just by sprinkling some of this compound on top of their dried distillers grains every day. So this effect is all well and good. Yes, it works. That's wonderful. However, we can't afford to go buy purified biocanin A and sprinkle it on top of our dried distillers grains every day. It's not economically relevant. And so looking at ways that we can supplement in this biocanin A and still see these benefits were of interest moving forward. So the next study we conducted was pretty similar. Again, we had some grazing steers. We had a pasture-only control and a dried distillers grain treatment, same as before. And then we had a low red clover and a high red clover supplemented treatment. And so we use red clover hay as the delivery mechanism for our biocanin A in this case, something a bit more economical. And so the low red clover treatment was about 15% of the total diet, and the high red clover was 30%. And so 30% would be pretty typical of what you might achieve if you were cross-seeding red clover out in your pasture. And so what we found was really interesting, not what we expected. And so the 30% treatment, that high red clover treatment, did not provide any added benefit to gain over traditional protein supplementation. But the 15% treatment, the lower level, impacted gain by 24% just by adding a smaller amount of red clover. And so this was confusing to us at the time. We thought, wouldn't more be better? Not in this case. More was not better. And so that kind of fits the functional food type of definition, right? Something lesser in the ration that can impact performance. As part of this study, we looked at our rumen again just to see what was happening in these animals while they were grazing. And what we found was that both treatments reduce hyperammonia producing bacteria, and that was good. And we didn't see any effect on our fiber utilizing guild. So again, good evidence for biocanon A being a good opportunity. But we did see an improvement in hay digestibility over these treatments, and the low red clover was better at doing so. And so again, this concept of a lower level being most effective. So there's been a lot of other research that we've done in this area, but some of the questions that we've run into and have been working on are things like isoflavone dose and source. And so when I give this information to producers in our area, typically what I get asked is, can I use a different legume? I have soybean meal access, or I might want to put some soy hulls in my diet, or I have white clover in my pasture. Will it do the same thing? And so we have some ongoing research looking at different isoflavone sources as well to see if we can gain those same benefits. There are always other production system type applications that can be used or supplementation strategies. Things like ear implants with isoflavones have been considered, and there's some researchers working on that, as well as feed forms. So looking at pelleting red clover so we can include it in our supplement every day to be able to attain these benefits. So when we were getting through this beef cattle research at the University of Kentucky, we have a really outstanding sheep unit and an outstanding sheep nutritionist, Dr. Don Ely. And he came in and he said, why aren't you doing this in sheep? There's no business like the sheep business. You need to be doing all of this research in sheep. And so we planned a 5-year project plan. I hope I have some small ruminant enthusiasts in the audience. That's what we're going to be talking about for the rest of the day. So we have this 5-year project plan just evaluating these same technologies for use in sheep. It's really new. So we're in year 2 of working on that, year 1 of the project plan. There's still 4 years to go. But today I'm going to share with you some of our preliminary work moving towards application of these isoflavones in sheep production. Keep going the wrong way. So why sheep? The United States is not a particularly large sheep producing country, but it does have substantial economic benefit. And so the total U.S. impact as of 2017 by the American Sheep Industry Association was about $5.8 billion, with direct impacts of sheep and sheep-related products of about $2 billion. And so anything we can do to improve that production, and also I think small ruminants are a really attractive option for grazing systems, and so maybe provide benefits that would increase the use of small ruminants in the United States would be excellent. So the first study we did, unfortunately, was not on pasture, but I want to share with you just to lead you through our story. We had 24 October-born ram lambs, specifically of the polypay breed, and so as sheep are very variable in their breed, so this was just in polypays. These rams were about 30 days post-weaning. They underwent a 2-week adaptation to the control ration, and so the control ration contained 85% concentrate and 15% forage, and then were assigned to one of 3 treatments. They were either given the control ration that just contained orchard grass in that roughage component. The next treatment was a 7.5% orchard grass and red clover split, and then a 15% red clover diet. So as I mentioned, all treatments were 85% concentrate, so this was more of a high carbohydrate type diet, and 15% forage. The concentrate was corn and dried distillers grains, and so those were adjusted to make the diets have the same amount of protein and the same amount of energy, and so that wasn't necessarily impacting the results. The roughage component was substituted out in order to get these red clover treatments of 7.5% and 15%. And they were allowed ad libitum access to these diets, and with the concept that we could look at things like feed efficiency. So here's some of the results from the study. And so how it's broken out here, we have average daily gain as part A, daily feed intake as part B, and then our feed conversion ratio as part C. I've split this out into 3 different periods. The first set of bars is day 0 to 28. And so the first month of the study. The second group of bars is day 28 to day 56, or the latter month of the study. And then finally, we have the whole study put together for each of these variables. And so the reason why I split out these time periods is so these sheep are rapidly growing from day 0 to 28. And you can see here that we didn't really see much of an effect on feed intake or feed conversion ratio in this period. However, when these rams were getting larger, and typically where we see intake go up and growth go down, we saw that the red clover changed that. And so we saw in the control treatment that lower gains and greater feed intake than in the red clover treatments, resulting in a difference in feed conversion ratio. And so if you just look at day 0 to day 56, because of this second month of benefit, we see an improvement in average daily gain. We see a reduction in dry matter intake with red clover inclusion in the diet at both levels. And this resulted in 25% less feed per unit gain needed over the period. And so a pretty substantial reduction in terms of what your inputs are to get the same performance benefit with both 7.5% and 15% levels. We looked at the rumen in these sheep as well. And when we didn't have red clover supplementation, we had more HAB present in the rumen. But with red clover supplementation, again, we saw a reduction in these hyperammonia producing bacteria, same as what we saw in cattle, this benefit that was linked to this change in rumen function. So the next question that we wanted to ask was, okay, we're down at 7.5%. We're lower than we were even at in those cattle grazing studies. Can we go any lower? We're looking at a functional feed. How small of an amount can we have in the diet and elicit the same benefits? And so we have ongoing research looking at that. And so year 1 of this experiment has been completed. And so I'm going to share with you the results from that. And year 2 is ongoing. Like I said, most of this research is ongoing. As part of this study, we looked at the rumen microbial community and ram lambs. And what we found was that when we added 7.5% of the diet as red clover, 5% and 2.5% of the diet is red clover. At day 21 of the feeding study, we saw that all reduced hyperammonia producing bacteria. However, at day 42, the 2.5 treatment was no longer effective. And so this leads us to believe that sweet spot in sheep is probably between 2.5% and 5% red clover in the diet of what we can include to elicit these benefits, or the lowest level. And so if you look at total intake of a ram lamb that's consuming about 4% or a little over 3% of their body weight, they're eating about 4 pounds per day, you would need about 1/10 to 2/10 of a pound of red clover per day to be able to elicit these benefits. So this study is part of a larger study. So there's a lot to come next on this study, like feed efficiency data to tie and see if we get the same benefits with a reduced HAB in these lower levels. And then also we've expanded to look at some carcass characteristics. So things like carcass yield and quality, meat quality, as well as some meat color stability characteristics of this lamb to see if there's some impacts of isoflavones on those measurements. Probably everybody in the room wants to hear, what about pasture? And so another initiative related to this 5-year plan is evaluating these supplements in pasture systems. And for this study, we just completed the first year, and so that's what I'm going to be sharing with you today. In this year, we had 40 April-born wether lambs, again, about 30 days post-weaning. These animals were put on orchard grass pasture for 2 weeks as an adaptation before we collected their first body weight. And then they were assigned to one of 4 treatments. They were either on that orchard grass control with no red clover supplementation, a 7.5% red clover treatment, and a 15% red clover treatment, similar to what that first ram study that I talked about in terms of supplementation level, and then a 40% red clover treatment. And so we have an interseeded red clover treatment in this study as well to evaluate those effects. The diet consisted of grazing plus 2% of their body weight of these different supplements. As part of the study, the animals were taken to a slaughter weight of 120 pounds and then harvested to look at meat quality characteristics as well. Taking a closer look at some of our grazing management related to this experiment, it was a 3 years grazing study, as I'd mentioned. We'll be completing or finishing this in 2025, so 2 more years to go. Each pasture was divided into 3 subsections and a sacrifice area in the front of the pasture, and we're using rotational grazing. In Kentucky, we have a lot of problems with stomach worms, and so this is really the best way things that we can do to control that. Each year, there's 2 pastures assigned to each treatment. with 8 animals included on each pasture. And so we have 5 polypay wethers that are our testers or animals that we're looking at for performance benefits. And then we have added grazers in each pasture. And that's just to optimize the forage yield and the forage stand to make sure that we're having good and adequate forage for these sheep. Again, these pastures are rotated every 14 days. So looking at some preliminary results, and I want to emphasize this is preliminary, year 1. So when we looked at hyperammonia producing bacteria on day 28 and 56 of these supplemented treatments, we saw that all treatments reduced hyperammonia producing bacteria. And then if we look at average daily gain from this first year, we got about a little over a half a pound per head per day, which is pretty typical for these polypays out on orchard grass pastures. And we saw that improved anywhere between 0.6 to 0.7 of a pound in our red clover treatments. And so we look forward to seeing what comes in the next couple of years, but there's some promising results to start. So stay tuned. There's several, like I mentioned, ongoing experiments that have a lot of different measurements that hopefully we'll have more information soon on. In sheep, we're also looking at isoflavone dose and source, and a similar method is what we're attempting in cattle. Supplementation strategies come up a lot. Sheep don't really enjoy our ground feed that we provide them in the trough, so we need to look at things like pelleting to be able to supplement this red clover in the diet. And then also one of our initiatives that have come up a lot from talking about this is isoflavone levels in the pasture. What factors influence those? And so I wanted to share with you as the last part of this presentation relatively quickly some of our research looking into the forage itself and how we might be able to influence isoflavone levels. First off, we have an enormous study that's just starting this past year and going for several years, our isoflavone national survey. And so we have several collaborators across the eastern United States that are submitting red clover, white clover, alfalfa samples, and also some regionally important legumes like peanut and crimson clover. And so you can see that there's a pretty good distribution of people participating in this study. And so the goal of this study is to look at things like species and variety of these legumes and how those might impact isoflavones, as well as plant characteristics, seasons, weather, and also location. So if we can identify some variables that might impact isoflavone concentrations. So we have some preliminary results from this experiment. So this is just data from our Kentucky legume variety trials. And so we have a pretty big variety trial program at Kentucky, and red clover is included in that variety trial program. And so this is looking at biocanon A across 6 different varieties here this past year. And so if you look just for the holistic view of it, there's a great variation between variety in terms of their biocanon A concentration. Bigfoot might be a little bit less than 2,500 micrograms per gram on average, while something like Blaze is up above 3,200 to 3,500 micrograms per gram. And so if you think about, is it not being about red clover, but the isoflavones that are present within it, the amount that is being delivered via the forage plant would influence that benefit. And it's possible that selection of particular cultivars might be beneficial. Another thing that we've looked at is a little bit on maturity effects. No surprise, much quality characteristics. Isoflavones are lower as the plant matures. And so you can see here biocanon A from vegetative to early seed pod goes down pretty dramatically in red clover plants across all of these cultivars. So the last thing I want to leave with you is just the concept of a functional feed versus a traditional feedstuff. And so when we think about red clover versus alfalfa and think about this concept, they have a major difference in isoflavones. So just from an isoflavone perspective, red clover has a lot of them and alfalfa has virtually none. And so typically in this context, when we think about alfalfa, it would be classified as a feedstuff. Even though it's the queen of forages, it's still, although a very excellent feedstuff, is a feedstuff, while red clover is a functional forage. And so something maybe we can strategically utilize in the diet to provide these benefits. So thank you guys for all of your time, and thank you for listening to me drone on about isoflavones for this entire time. Obviously shared an enormous amount of information. I wanted to give you guys a broad overview of everything that we're doing. This is my contact information. You're welcome to ask me questions after Dr. Brito's talk, or feel free to contact me at any time if you have something related to your system or your research program, or in any case, any question you might have, I'm happy to answer it. Thank you. Would anyone like to ask some questions now? I think I've left a few minutes, sorry.
But yeah, I think we have time for just one or two questions before Andre. Andre, maybe I'll introduce you now so you can start getting set up. And so we have Andre Brito is presenting next in this really informative session. That was really great.
Thank you.
So yeah, we have time for a couple of questions now while Andre gets set up, and we are going to try and get the mic to you so our friends online can also hear the questions. Nico had one. He was jumping to
Chomping at the bit, yes. So I'm going to try and summarize that. I assume you controlled for maturity in your isoflavone. The first quantity assessment of the different varieties of red clovers were controlled for maturity level of the forage samples.
So the question, I guess the question is
Yes.
about the mag, so that's good. Actually, it was just averaged across all of those maturity samples. And so it was an average from every sample that was taken off that maturity.
Well, yeah,
line.
it wasn't
So-
quite clear on because there seemed to be a big variability potentially introduced into that. And then the sort of overarching question is, you don't really know why the higher isoflavone, or in this case, red clover samples produced lesser results, right? We're in the dark about that.
We don't know, and we don't have we took rumen samples as part of that study to try to be able to tie that with those gain effects we were seeing, but we saw nothing in the 30% treatment that elicited any concern about a negative effect. We would guess that it's probably like an upper amount of phytochemical or an antimicrobial in the diet that might be having effects on the rumen microbiome that we could not measure in the measurements that we took. Sometimes when we add in antibiotics into these bacterial communities, we can have a negative effect by killing too many things. And so possibly we just exceeded that amount.
Yeah, or it might have nothing to, this sort of follows a discussion with Juan Alves from Extension Vermont over the last, I don't know, 8 years of what is the optimum quantity of legumes in a pasture mix. And so there might be correlations there to that quantity as a whole.
Yes.
With the trend being get legumes give you better results.
Yeah, and so
Or in general, and it might not be that's entirely tied to red clover, but it's very interesting that there's that intersection there.
yeah, and the really interesting thing about, obviously all of these plants contain many different phytochemicals. So we're just talking about isoflavones and biocanon A today. But if you look at different legumes, the profiles and the quantities of these compounds are vastly different. So if you look at red clover versus white clover, both clovers, both in pastures, white clover has 200 times less total isoflavones than red clover. And so the amount that you include in pastures gets very complicated when you think about it that way. You got forage maturity, you have variety, and then you have species, right? And you have all of these enormous differences. Like I mentioned, alfalfa is very low. Soybean meal has a completely different profile. And so will it provide the same benefit? I don't know. We have some research to look into that, but it's an ongoing question for us too. Discussion point. Yeah. Yes.
I think we have time for one last quick question, if anyone has any. Otherwise, we can wait until the end after Andres.
Burning, I would suggest that we wait till the end
Yep,
to make sure we give
Yep.
Dr. Brito enough time.
that sounds good.
Cool, thanks.
Thank you, Andre. Take it away.
Yeah, thanks, Damon, and thanks for the organizers for the invitation, Jen and Megan. Yeah, so follow up some of the conversation here on phytoestrogens as well. We have been engaged in some of that research at UNH. Just the outline of the presentation, looking at the importance of forage and forage legumes, right, in dairy diets. I'll be sharing data from a study that has been recently published where you're looking at alfalfa versus red clover based diets on several parameters, including milk fatty acids, nutrient utilization in dairy cows, and then some of the environmental factors and management factors that can impact the accumulation of phytoestrogens in forage, but with the focus on red clover, right? And then some conclusions from this work. This just starting related to the importance of forage, right? This is data from a recent survey. The paper has been accepted, and that's led by Heather Darby lab in UVM. We're looking at surveying the organic dairy farmers across the US, and what they indicated is, it's what I guess we all agree, is that forage, right, it's important, somewhat enhancing or significantly enhancing milk production body condition score, reproduction, and calving, good for herd health, quality of the young stock, and also for farm income because you can reduce grain costs. We also looked at the satisfaction of the organic dairy farmers in terms of the forage production systems across the US. And in general, they were somewhat satisfied with the pasture yield and quality, storage forage yield and quality, less in terms of legume content and persistency. So there is some work that could be done there to improve persistency of in pastures. And this is another data set from a study that in this case was led by Kathy Soder right there. And Amy Heffler was a postdoc that looked at this set of data from farms across the northeast of US, from Maine to Vermont, New Hampshire, Pennsylvania, and New York. So we collect samples, pasture samples over the years, 3 years, and also looked at botanical composition. And you can see, as expected, there's some variation in the proportion of legumes in those pastures, right? From as low as 22% in 2012 up to 37%. But the point here is that we're trying to understand how much legumes are on those pastures in the context of their importance in terms to improve milk production. And this is data also from UVM, a seed buzzword, what they looked at trying to see what the threshold about how much legumes should be in the pastures. And they indicate that roughly you should have at least, I'd say 30% of legumes in those pastures. If we increase that proportion, you start to see less fiber, right? Less NDF, less ADF, and more crude protein. And this usually correlates with more intake. As you decrease fiber in the diet, the cows can consume more feed, and that can translate in more milk, right? And looking at this grass and legumes, I'm not advocating here to not having grass on pastures, obviously. But the point here is trying to highlight the importance of legumes, right? And this was a meta-analysis that was published some time ago by folks from Europe, but look at data across, right? Across the temperate climates, where they compare diets that has grass, and this mostly silage, versus legumes, right? And what they saw in that meta-analysis, legumes results in more intake, right? So usually a 6 to 7% in intake of dry matter, more milk production, and more NH-corrected milk production. So that means that probably the milk components also was higher, like milk fat and milk protein, despite a decrease in digestibility. Organic matter digestibility decrease. So it indicates here that the rate of passage of legumes in the rumen is faster than grasses. So they move fast. It's less digestible. The cows consume more. So that's counteract the drop in digestibility. Again, that's highlights here the importance of legumes. And then in the same study, they break out, right, the different types of legumes. And also they did for grasses, but I'm just showing here the legumes. And either white clover, red clover, alfalfa, bird's foot trefoil. And in general, you can see that both white clover and bird's foot trefoil, but specifically bird's foot trefoil, it did increase dry matter intake, produce more milk and more NH-corrected milk compared to red clover or even alfalfa, and even superior to white clover. Statistically, as you can see, it's not different, but numerically, you can see that's a big difference in some of those traits there, in production traits. And then look only at red clover and alfalfa, which is the focus of this talk today here, at least in one of the studies that we're showing. Oftentimes, those are equivalent, right? You can see in terms of intake, milk production, and NH-corrected milk, they're relatively similar. But one thing I have seen in a lot of studies, and also here in this meta-analysis, which basically combined data from several studies, was that red clover often results in diets that are more digestible. So your organic matter digestibility was higher here in red clover, that with fiber, NDF, ADF, but this not often translate into more milk. So you would expect, okay, more digestible, more energy, more milk production, and that doesn't happen with red clover. And you try to understand energy partition in diets with red clover versus alfalfa. So that's where showing some of data here on that regard as well. Going back to that survey across the US, we just trying to understand what the proportion of farmers grow different types of perennial grasses or perennial legumes. And not surprised here, 83% of those farmers, they do have orchard grass on their pasture mix or in their hay fields. And this survey was more populated with farmers from the Northeast region. So that can explain maybe the high level of orchard grass in those farms. And in terms of legumes, you can see that 87 and 86% of those farmers indicated they do have those legumes in their fields, right? That, of course, highlights the importance to be looking at red clover also as important legume. One thing that's about red clover, that in addition to the phytoestrogens, red clover also has this other bioactive compound called polyphenol oxidase, or PPO. It's an enzyme that basically phenols to quinones in the presence of oxygen. And ultimately, what it basically does is protect against proteolysis or degradation of proteins. During siling, if you make a silage of red clover, you have less proteolysis, less breakdown of proteins. And this translates oftentimes in cows having better efficiency of nitrogen utilization, right? So that's another aspect of red clover that that's important to consider. And in addition to inhibit the breakdown of protein, it also inhibits some enzymes as well, like lipase that can break down fatty acids and things along those lines. So when you look at red clover, right, so one thing that's consistent, right, is it reduced ammonia formation in the rumen. Is that associated with the biocanine, probably the hyperammonia bacteria? But if you're talking about silage, it's probably associated with that PPO enzyme that basically reduced the proteolysis, the breakdown of protein, so there is less ammonia formation in the rumen in those cows. So with less ammonia in the rumen, you usually have less urea, right? Less nitrogen being excreted to the environment. A proxy to that, to look at that, is MUN, or milk urea nitrogen. When you see diets with red clover, you also see that lower levels of MUN, or milk urea nitrogen, indicating that improved nitrogen utilization in those animals, right? That's give you a little bit of the background why we're looking at red clover. And this was a study that I'll be sharing some data, just to keep in mind that this was diets, high forage diets with baleage, right? And this was fed to organic cows. So just very briefly here, the nutrient composition of the baleage, you use two types of baleage, two different cuttings, either alfalfa or red clover in second and third cut. What I'm going to show here is what I said, soluble protein. You can see that's much lower in the red clovers compared to the alfalfas, right? And that's primarily driven by that enzyme polyphenol oxidase that reduces breakdown of proteins during siling. Another aspect that to look at here, other than that, you see the NDF, ADF, ligninos, all similar. But red clover seems to have, as compared to alfalfa, more sugars. You can see the data there, and while alfalfa seems to have more starch, right? So in terms of the non-structural carbohydrates, so more sugars for red clover and more starch for alfalfa. And that's the diets. We fed a 65 to 35 forage to concentrate ratio, the same grain blend of the concentrate, and then what vary in these diets, either alfalfa or red clover. And you can see some of the nutrient composition of the diets. It was relatively similar in terms of EF, crude protein, and that. So, series of samplings on the, across time. We have an adaptation period, and then series of samplings on 4 weeks after we start the study, and then on week 7 after we start the study. This was a study that lasted about 7 to 8 weeks. We did collect gas emissions of these animals. The goal here is not feeding red clover or alfalfa to mitigate methane, but to try to understand energetic losses. Methane represent energy losses by the animal. We use the green feed system. I think, I'm not sure if folks are familiar here, but it's basically a new, not too new now. Technology has been out there for maybe 15 years now, close to that. It basically you have a bait, the cow's going to trigger the system, it releases a bait, and then the cows are consuming and breathing and belching. So there is sensors here that can measure methane, CO2, oxygen, hydrogen, so you can have an idea in terms of the gaseous emissions from these animals. In this study, also collect rumen fluid, milk production, milk composition, urine and feces and blood. So in this way, you can estimate urinary excretion, digestibility of nutrients, collating ammonia, amino acids, things along those lines. And for the rumen fluid, we analyze for ammonia and the VFAs, the volatile fatty acids that Brittany was talking about here. So here's some of the data of this study. Did not see any effect on dry matter intake or milk yield difference. However, energy-corrected milk was higher in alfalfa, and also feed efficiency, if you express here in milk production over dry matter intake, those are higher in alfalfa. And this probably associated this increase in energy-corrected milk with the milk fat. We did not see a difference in milk fat percentage or milk protein percentage or lactose percentage or some of those milk components, but milk fat yield, right, tend to be higher in the alfalfa diet. So that pushed that energy-corrected milk to be also higher. We look at the milk fatty acids, and as I said before, this enzyme polyphenol oxidase, it tends to block the lipase, the enzymes that break down lipids, and that reduced the biohydrogenation processes that takes place in the rumen. In other words, it preserves more of the unsaturated fatty acids. And what you can see here is that the alpha-linolenic acids, the major omega-3 fatty acids, so the total omega-3 fatty acids and that omega-6 to omega-3 ratio decreased. In other words, red clover had result in milk with more omega-3 fatty acids, and also with a better omega-6 to omega-3 ratio, right? The lower, the better we know was related to human health, right? And this is also an important aspect when you look at human health and things along those lines. In terms of nitrogen utilization, as I said before, that enzyme polyphenol oxidase, or maybe some of these phytoestrogens, maybe together here. As I said, several studies have been showing, we confirmed this again here, that red clover usually reduced, right? So those MUN or plasma-urea nitrogen, urinary excretion to the environment, ruminal ammonia nitrogen, you can see very clear here. And that, again, is important when you look at the environmental impacts of dairy systems. There's some forage by week interactions, and see most of the response happened during week 4 and last during week 7. But overall, right, you see this reduction or improvement in nitrogen use efficiency. In terms of digestibility, again, so this happened also in this study. You can see that digestibility of dry matter, organic matter, and fiber and NDF all higher in red clover diets, but that was not translating to more milk production. In fact, energy-corrected milk did decrease when the cows are fed red clover. So we're trying to understand what's going on in terms of the energetics, right? So this is the typical energy partition in diet. So you go a lot from gross energy up to net energy, and then there is energetic losses. The animal is going to lose energy through feces, right, to gas, primarily methane, urinary, and primarily nitrogen out of the urine, and also the animals generate heat during digestion processes. That's the heat increment. So we can use the calorimetric bomb to measure some of those energy in some of those matrices, milk or feces or urine. We have been using the green feed, right, to estimate, to measure methane, also CO2 and hydrogen and oxygen. You can estimate your energy. We didn't this study with this equation, but you can also use the calorimetric bomb to measure directly. Heat production, we measure with this equation, rely on carbon dioxide, right, emissions, and this coming from the green feed system. And then you can measure net energy intake, or estimate net energy with the NASEM, the new NASEM equation, with the old NRC. And then tissue energy, you can estimate by subtracting metabolizable energy from heat production, milk energy. So you get metabolizable energy. You get, back calculate to get tissue energy, and then milk energy you can estimate with the equation also from the exam. Using this process here is a mix of measurements and estimation through equations, right? There is some uncertainties when you do that, but that's the only way to do it. In terms of gas production, what you saw here is that there's an effect, still not completely understand this effect, but if the red clover diets did decrease methane production, and this was a little bit surprising in the sense that there's no effect on intake. A little bit difficult to explain this response here. But when you looked at methane yield, which is grams of methane per kilogram of dry matter intake, or methane intensity, it's grams of methane per kilogram of energy corrected milk, there's no difference there, right, per kilogram of product. Not going to spend too much time in this table here, but just you can see the breakdown of the energetic calculation and highlighting some points here. In the case of red clover diets, that has less fecal energy because the diets are more digestible, less urinary energy because they excrete less nitrogen, as shown earlier, and less methane energy because they produce less methane. And another two aspects here was that milk energy was low in red clover because less energy corrected milk, less milk fat. And tissue energy, that's what I think is some interesting here. Tissue energy, that's retained energy in the tissues of this animal, seems to be higher in red clover. It indicates that some of this energy partition is, rather than going to milk production, it seems to go into to accumulate body tissue. And this, you can calculate this through efficiency. You can see in this case here, that's metabolizable energy. Cows on red clover, they were less efficient to convert metabolizable energy into milk energy, as you can see here, primarily during week 4 of the study. And they were more efficient, or maybe less efficient, to convert metabolizable energy into tissue energy. In other words, the energy partition seemed to be going towards more accumulation of tissue rather than to milk production. I don't know the mechanism behind this, but that's what some of this data is indicating. So I think we need more studies to look into this energetics of forage in general, right? So switching gears here now to the phytoestrogen conversation. So Brittany was discussing, right, primarily about the flavonoids. I also have other classes of phytoestrogens, the lignans and the stilbenes. I don't know how to pronounce this, but anyways, they're difficult overall. The isoflavones are common in some of the clovers that we just discussed, and what's the focus here of this conversation. What I want to say here, these phytoestrogens, now the name is there for a reason, right? So you can see the molecule of estrogen there, and genistein is a type of phytoestrogen. They structurally very similar, and this could have some negative effects, right? There's some positive effects, like Brittany discussed, but it's potentially some negative effects associated with phytoestrogens. And some of them are here. And you can see this data, lots of review was looking at in the human literature. You can see the phytoestrogen has been associated to be beneficial to reduce menopausal symptoms in women, right? Reduce cardiovascular disease or obesity, type 2 diabetes, improve brain function, reduce cancers. But you see all the question marks there because there is no consistency or not enough data. On the other hand, there is potential negative impacts on human health from endocrine disruption. You can reduce thyroid function, reduce brain function. Some breast cancer, in fact, could increase. So there is like a little bit of a black box there where we don't know if the benefits outweigh the risks or not. You basically need more data on this phytoestrogen story here. So again, when you look at the animal itself, and this, I was in Australia this past summer as a part of the visiting some university and discussing about phytoestrogens, because they have a lot of issues there with the sheep, primarily associated with reproduction issues, infertility. Their clovers, they have different types of clovers that have the high levels of these phytoestrogens as well. And here's the general metabolism of isoflavones. In fact, in the animals consumed, there is this extensive metabolization in the rumen of some of these compounds, producing one of the major ones, the qual. It's highly active, one of the more bioactive. That's what you're going to potentially find in milk that you're drinking. And of course, some of this equal can circulate in the blood and potentially, right, mimic estrogens, but they don't have that. They have weak or strong effect, and depending on how those interactions take place, it can eventually disrupt reproduction, cause infertility, cause embryonic losses, but there is not much data on cows. There's some data on sheep, on cows. We don't know much about it. So these are the potential negative effects, and there is potential benefits, right? As Brittany discussed, increasing growth rate, weight gain, improved nitrogen utilization. So we need to better understand, right, where we are in terms of this phytoestrogens situation. And here is an example of a study that was done, I think in Norway, where they collect milk samples and buy milk samples, retail milk samples, organic and conventional. And you can see pretty clear here on the top line there, that's organic milk, right? In general, organic milk has this higher level of phytoestrogens compared to conventional milk. And this potentially associated with organic, right? More forage in the diets or pasture-based diets, more clover intake. Conventional, we know, is more TMR-based. You have less forage and maybe less grains that would cause some of these effects. But you can see that equal, for instance, it's very high levels compared to the other phytoestrogens that indicate that a lot of the, for instance, biotin A, not biotin A, but formononetine, which is another phytoestrogen, is converted into equal, and daidzein also converted into equal. And we don't have data like this here in the US yet, as far as I know. We're looking at, actually, me, not necessarily, but my student, and I'm co-advising with Rich Smith. And Palash has done a lot of studies looking at factors that can impact phytoestrogens, right? That is agronomic factors, forage cultivars, like Brittany was discussing here, the biotic, abiotic effect, insects. So he came up with a set of studies for his PhD. One of them was looking at insect herbivory, how insects could potentially impact the concentration of those phytoestrogens. And he looked at red clover and cured clover. It's supposedly to be no phytoestrogenic. That's some gray literature showing that. Now we have some data on this, but what they show in this first study, they show a species by treatment interaction, meaning you can see that this increased response, right, in formononetine, which is one of the most important phytoestrogen in clover, and also biotin A, when the insect promote that damage in the leaves, right? So phytoestrogens is a way that the plants use also to, as a mechanism of defense, is an antioxidant for them. So they are defense mechanism. When they're stressed, they can accumulate those phytoestrogens. And you can see here, if stressed by a insect, it can cause increase in phytoestrogen over 100% here. You can see the formononetine, the biotin A, and the total phytoestrogens there. So it has been some data showing that levels of about 3 milligrams per gram of phytoestrogen could be dangerous for sheep. We don't have data for cows. Dangerous, what I'm talking about here, mostly in terms of affecting their reproduction. He also did another study where he looked at the effect of drought and waterlogging. And this, again, this is on the greenhouse studies, where they have a control, basically 100% water holding capacity, and they have a drought, which is 40% water holding capacity, with 20 days of that, and then 10 days of drought only. Either waterlogging for 20 days and waterlogging at 10 days. Again, it's trying to promote stress in these plants. You can see here in the middle, that's the control plant that was supposedly in the best conditions. You can see, then you have in the left side would be the drought, my left side. And then in the other, the waterlogging, you can see that the plants are affected. So he did see an effect on the red clover in this study, and a huge increase, again, with the drought, 20 days drought. It caused about 100% increase in the total phytoestrogens concentration. There are no phytoestrogens in cured clover in this study. We were able to find very small amounts in the first study, but no phytoestrogens that was detected, let's say, detected by the instrument, right? In terms of the other phytoestrogens here, the formononetine and biotin A, so you can see those are the total. Now we'll break them down into the species. You can see the right there pulling this up on that 20-day drought treatment. Again, that's certainly above that level that's considered safe for sheep. And then when you look at the other ones, there's minor phytoestrogens that are also present there, like daidzein, for instance. You can see that one kind of interesting that it responds to waterlogging, it increased, but not with the drought. And the other two minor phytoestrogens here, you can see the same, it responds to both, right? So either drought, 20-day drought, or the waterlogging, either 20 or 10 days, cause that increase. That's interesting. It seems to, depending on the type of phytoestrogen and the stress, the plant responds differently. Just this showing that in red clover, the most of the phytoestrogen that you're going to see there, independent of the stress or not stress, is either it's both formononetine in green there, and that biotin A is the one in kind of a gold color there. So it's quite clear those are the two most important, and when you're looking at ways to manage, so those ones will be more in line. And just to finalize here, we also look at the effect of cutting management. This was done in the field, in the plots. The treatment here was basically, we have legume grass mixtures, right? We have either orchard grass, white clover, or orchard grass and red clover treatment. And you have either intensive cutting, was 5 times a year, versus less intensive, 3 times a year, in different heights at 5 or 10 centimeters height. That was the treatment. The height, cutting height did not affect the phytoestrogens. However, the frequency did affect. The more, and this is a total phytoestrogen, so the more intense the cutting management, it increased the phytoestrogen significantly. The total in white clover, there is no effect. And you can see, if you see the y-axis, like Brittany was discussing, and of these two graphs, that you can see that concentration of phytoestrogen in red clover is much, much higher than that in white clover. And then when you look at the other phyto, for instance, basically now speciating them. So formononetine was the one that caused that increase, and biotin A it increased, but not statistically. And the other two here, zine and the glycine, these two, it decreased, right, with the more cutting. Again, that's a little bit difficult to explain here, but it seems that the mechanism behind controlling this is not fully understood, but it seems that it does affect one more than the other one, depending on the management that you're applying into the system there. And then to summarize, so we have some time for questions, compare with grasses, legumes, based on that meta-analysis, it does increase dry matter intake and milk production, despite a reduction in digestibility. So in other words, that increased dry matter intake counteract, right, the drop in digestibility when you have legumes. Compared to alfalfa, the red clover improve nitrogen utilization, nutrient digestibility, omega-3 fatty acids in milk, but did decrease energy-corrected milk, and this may be associated with that energy partition that we discussed. The phytoestrogen in red clover increased, right, in response to stressors, and either for insect infestation to drought, waterlogging, all of them was able to increase those levels of phytoestrogens. But it's important, more research on energy partition in forage, right? In general, there is very sparse data in that system, and also trying to understand the mechanism of this phytoestrogen response so that we can change, but what is regulating those change, it's not, it needs for sure more research. So I'll acknowledge the funding source of this project, USDA and Northeast SARE, and New Hampshire College of Life Science and Agriculture, the organizers for the invitation, and I would open to questions.
Great. Thank you, Andre. Why don't we, Brittany, do you mind coming up here as well, just so you both can be grilled? That was really two great, very informative presentations. So thank you both very much. Yeah, we can start with, and actually, I will give you this one.
Hello, good morning. Thank you very much for the presentation. My question is related, do you, knowing that the situation with that phytoestrogens and the sheep, do you plan to have any research related to how those stressors in the clover could be impacted the reproductive performance of the sheep?
Yeah, it does impact we're trying to understand now at UNA, not myself, but there is another researcher that is looking at the ovary function and corpus lutein function associated with phytoestrogen exposure. What I can say, and based on what I learned in Australia, it is an issue on several farms. I visit, I don't know, maybe 5 or 6 farms in Australia. It is an issue. However, of course, they are managing this over time, right? They try to have varieties that are lower levels of phytoestrogen. They have subterranean clover, which is very common there. They're trying to get away from some of those and trying to manage that in that way. But it does have an impact in reproduction efficiency on sheep, and I expect to have in cattle. However, the levels, right, or the exposure and the time that could lead to those negative response, I'm not totally sure about it. What I heard from some farmers in New Hampshire, a couple of them, actually they contact Pete Erickson, which is the extension, you may know him, and a couple of farmers contact him with issues in reproduction, cows not get pregnant, and they narrowed down to red clover. They have very high levels of red clover on pastures and on the storage forage. But those are somewhat, anecdotal, right? I think certainly studies are needed to be done, primarily looking at the molecular level. My colleague is doing now at UNH, and also large, maybe doing reproduction studies. It's complicated because you need large number of animals. It's not always easy to do, but the problem may be there. Yeah.
All right, great. Other questions? You got one in the back here.
Hi, thank you. This question's for, is it Brittany? So your presentation was one I really wanted to come and see as a farmer. That's the data that I need. And I was curious with your current research where you have the 3 paddocks, the N of 8 plus the 2 extra grazers and the 14-day rotations. I was curious because we rotate sheep. So my thought is when I set up a paddock, 8 sheep is a tiny group, so there's not much competition for food. So I was curious why they're grazing for 2 weeks. Because if in our experience, if I'm trying to get them to eat everything, I move them every day because it forces them to compete for the food and I get to control what they're eating. And if I leave them in a paddock for 2 weeks, it's enough time for things to regrow and then they can pick and choose. So I was curious as to why that's designed that way.
Thank you for pointing out the #1 limitation of our sheep research study. Yes, you are 100% correct. So one of the limitations we have is the number of animals that we can utilize in these experiments from our sheep herd, or our sheep flock, I'm sorry. In April, we're limited on the number of polypay wethers we can use. So we're grazing with a really small group. So we are not seeing proper utilization of those paddocks. So 2 weeks essentially because they don't barely even capture the forage in those paddocks in 2 weeks. And yes, we would really prefer to be using a greater stocking density and also a faster rotation system. I certainly support that as a management tactic, but we just don't have the numbers to do that. And actually the reason why we don't have the numbers is because of the fertility impacts related to phytoestrogens. My sheep collaborator does not want to put any of his females or any potential rams on those pastures with the concern, especially with the interseeded treatment, that it would be too high. And so very good question. Unfortunately, that is probably the #1 limitation at looking at the impact of these treatments on pasture, because we're not really getting a good clearing of that pasture.
Great. How about other questions?
I think it's related to that last question as well. Again, thanks for your presentations. It was sort of towards the end of what you were presenting, Andre, where there was the change relative to the intensity of the cutting of the different phytoestrogens. And am I right? It was the ones that popped up on the right side were going down and the ones on the left were going up with higher frequency or more intense cutting. Was there like a relative level? It just seems like the cutting affects the profile, not just the overall levels.
Yeah.
And I'm just was trying to remember back to the pie chart and whether it was those ones that were overrepresented Like, how did that correlate with the pie chart?
Right there. You can see, yeah, you can see that. I think that the thing is the ones in the on the left side there, which is basically the, oops, there, let's put like this. These are the ones at very high levels, right? For mononitine and biotin A, those are made over 95% of the phytoestrogens in red clover bee that made up by these two. So they seem to be more responsive, specifically for mononitine A statistically, right? The other one is increased numerically, but to that cutting, management. The other two here is very minor. If you see the y-axis, there's a very small concentrations. And yes, in fact, those decrease, but for the overall response in terms of total, right, phytoestrogen is still going up because that mononitine A make up the bulk of those phytoestrogens. I don't know exactly why this happened in terms of why the cutting frequency would increase some of them and decrease the other ones. I don't have that answer.
And how that would affect the relative effect of those on the right that are decreasing versus the effect per the quantity. It may be a different effect. And so just looking at the profiles, just thinking about all the confusion between are phytoestrogens good or bad, or what are the effects? This might be in that profile and also related to intensity.
Yeah, this mononitine, if I'm pronouncing this correct. But this one is the one that's converted into equal. And that equal, when the animals consume this phytoestrogen, they get metabolized in the rumen, right? And for mononitine gives off to equal, which is a secondary phytoestrogen product, metabolite, that is associated with infertility. If you think about maybe potentially, in this case, would be more problematic because it's more for mononitine, potentially more equal circulating in the blood of those animals. And that is the one that's causing the issue with reproduction. In this way, it's more concerning for sure, the increase, not only the amount, but also the type of the phytoestrogen that went up.
I have a question over here.
Yeah.
Nico's monitoring the online chat. So he's got.
Yeah, so there's a few questions that got posted on the Hoover platform from people who have zoomed in basically. And there's their voice here. The first question is, I'm going to summarize a bunch of them in a way. At some point you talked, Andre, about the percentage that there was a reduction in methane production with which would correlate you guys in Kentucky, but it wasn't clear what the percentage of red clover was in the diet at that point. Could you elaborate on that a little bit?
Yeah, the 65%, that was a study where you have either alfalfa baleage or red clover grass baleage, right? There's some grass there in that mix. But about 80% of the mix was red clover. So the total diet has about 65% of red clover. So there's a very high level of red clover in the diet. Yeah.
And then it seemed that part of what your data incidentally showed in terms of the energy utilization between alfalfa and red clover, that it might be that one is more advantageous to milk production, the other one more for meat production, in terms of how an animal actually uses that, or these specific dairy cows. Obviously, that hasn't been tested out yet, but that would be an interesting hypothesis potential.
Yeah, I agree. Yeah, if one thing, want to make sure,
it's the same size as the south thing.
This NH partition is tricky and
And a lot more besides.
the whole thing, right? So it's a combination. When you do this, you take into account samples, urinary samples or fecal samples that you collect spot, right? So, and then you estimate the digestibility and urinary volume. There is some uncertainty here. And of course, we don't know the composition of that tissue. However, the hypothesis, I agree, right? So maybe if it's energy partition towards tissue more towards milk energy, and in red clover partition more toward tissue energy, hopefully muscle, then it'll be potentially more advantageous beef cattle, red clover and alfalfa too.
As a beef cattle raiser, we watch for the sugar and starch components very much, and there's a definite difference in effect of how weight gain happens there. So it wouldn't surprise me to see that bear out in some form?
Red clover does accumulate. There's some research from Canada. There's a lot of research with PM, AM, cutting at PM or
Oh, yeah.
having
Time
animals
of
grazing
day,
at PM, they increase sugars, and red clover was the one with the highest sugars of all legumes that they looked at.
So there's one more question here that looks at production and lifetime production, and that is something that I didn't see in this data. Maybe this is premature. So we're looking at ad hoc milk gains essentially of a fairly short period of time, but the diet that is high in protein, low in fiber might have life expectancy impacts. And so the lifetime production of a cow could potentially be affected with that. But what I'm thinking here, interpreting a little bit more than what was in this question, is how we know that CAFO operated, how operation, the life cycle of or life expectancy of a dairy cow is about 5 years. With grazed cows, it's twice that. And so maybe we can see some effects there of the digestibility to protein ratio, or fiber to protein ratio in the diet on life cycle. And so there, a cost analysis or an economic analysis would probably make sense to see pushing a production with high protein actually makes sense over the life cycle of a cow. Or if the cost of replacing heifers sooner, bringing replacement heifers in sooner would counter the effect of the temporary gain in milk production. Is that something that, could you comment on that?
If it's understood the question, that advocating for more protein in the diets relative to fiber, that was...
Well, when we looked at the samples of the forage, it was, we saw that with the higher amounts of either alfalfa or clover, milk production tended up, alfalfa more so than clover, but the typically dairy analysis is geared towards higher milk production. That's the goal. But the question, is there detriment effect from either the phytoestrogens or from the high protein in terms of the life expectancy of the cow, and what is the economic impact on that overall?
The thing is, diets high in nitrogen, right? Let's say in this study, you have diets that are 18% crude protein. That's too high. And the reason for that, because we want to feed a high forage diet. These levels of crude protein at 18-19%, you're not going to see in typical high output commercial dairies here in the US. You're talking about levels that are probably 15 to 16% with protected amino acids and things along those lines to compensate. The thing is, a lot of protein, you end up excreting a lot of those nitrogen, right? So that, as I show. So with red clover, it's clear a positive effect on the nitrogen side, right? It does reduce nitrogen excretion. So I would not think that levels of 18 to 19% would be, let's say, to a certain degree, impact negatively the health of the animal, but there is data showing that higher nitrogen, higher circulating ammonia, and urea could have negative impact on reproduction as well.
There's a metabolic cost to that.
Yeah, and then exact metabolic cost to detoxify ammonia in the liver with urea, right? To urea. So I would not recommend, of course, if you could balance diets with low levels. I'm a dairy nutritionist, that I would recommend. In studies, organic sector, in organic dairy farms, high forage diets, it's difficult to make diets that are low in crude protein. If you're talking about grass-fed sector, then you're talking about diets that are very high in protein.
So we do have one more question. We're running up against time here, so unfortunately we're going to have to cut it off after this one, but we do just have one more question.
I was just thinking back to one of the first slides that we saw with the Fred Provenza quote and the buffalo and the really diverse pasture, and just curious how unique the red clover is, or do you expect it is, as far as levels of these compounds? And if you don't think it's super unique, how would you go about, would you have any sense predicting what other species forages might have elevated levels?
That's a really good question. And there's some research, quite a bit of research, just looking at some traditional type legume sources like red clover, white clover, or some of what Dr. Ben. Red clover is particularly unique just because it's really high in these compounds. But we talked about there's probably optimal levels that might be lower and maybe would contribute to differences if you looked at lower levels in different production systems. And yes, I think there's other legumes that have not been investigated. There's not a lot of information about things like peanut regionally important legumes that might be present in grazing systems or more attainable in grazing systems that might be able to provide the same benefit. But we're hoping to find out a lot more about that moving forward. It's a big part of our next 5-year project plan is doing that. And so hopefully I'll have more information, but I do think that generally we just consider to be red clover a really rich source.
Thank you both so much. Let's appreciate our presenters for this round.
Okay, guys.
And so now there's time for a brief break, but Jen will be introducing our next session at 10:15.
Yes, thank you. It will be starting at 10:15. And I do want to encourage folks, even folks in the room, even if you're a person who doesn't want to ask a question out in front of the room, if you're on the Whova app, you can ask questions in that and we will ask them for you. So if you don't worry, if you don't think it's a great question, please ask them in there. Whova is great for both the online and the in-person folks. Thanks, guys.